let's talk about what epithelial tissues are before we get into transportive epithelia. So when we talk about epithelia, this is usually our lining tissue of the body. This is found everywhere that the body comes in contact with the external environment. So think about the lining of your nasopharynx, the lining of your GI tract, your reproductive tract. Your skin is an epithelia. Your entire mouth, the oral cavity, your tongue is covered by epithelia. All right? Epithelia is that tissue that lines or is a barrier of protection against the external environment. Okay? It lines your reproductive tract, your urinary tract, et cetera, et cetera. And so we want to speak about movement across this membrane because it's really important, right? Secretion of substances, absorption in the GI tract, the movement of water across the epithelium is important in terms of a lot of the function of these structures, okay? Now, it involves two membranes. We speak about the actual epithelial membrane. So when you look at epithelia, epithelia are nothing but a bunch of epithelial cells. So this is a single epithelial cell. And they sit next to each other, side by side, by side, by side, and they form that membrane. And so what we have is a membrane, but we also have an underlying membrane called the basement membrane. And then underneath the basement membrane, we have the underlying blood vessels. Now, epithelia are avascular, meaning that they don't have their own blood supply. They have to get blood supply from the underlying vasculature, and they have to move food or whatever is being absorbed from the lumen, so this will be the lumen of the GI as an example, through this cell and into the underlying vasculature, into the underlying blood vessel. Okay, and so this becomes really important in absorption of nutrients from the food we eat. It has to make its way through the cell. It can't go through the sides. We'll talk about why not, right? There are junctions here to prevent this paracellular movement. It has to go right through the cell and into the underlying blood vessel beneath. Now, we have different structures on different sides of the epithelia. On the apical surface, we typically have some type of apical modification, usually a microvilli. Microvilli increase the surface area to help us absorb more. We talked about surface area and absorption. On the lateral sides of these epithelial cells, we typically have tight junctions. And tight junctions are one type of junction that's found on the lateral domain. And they basically prevent movement of molecules or water through these paracellular areas. So movements of particles cannot happen through here. It has to go through the cell, through the basolateral membrane, out into the underlying blood vessel. Okay, and then the final or side of the cell or domain is the basolateral membrane. And this is the membrane that sits right on top of the basement membrane, which sits right on top of the vasculature. The basolateral membrane is pretty much the bottom of the cell or the base of the cell. This is where the cell goes up from, right? So we have a lot of mitotic activity at the base of the cell. Let's talk about some features that we find on different parts of the cell. So on the basolateral membrane of the cell, we typically find sodium-potassium pumps. We talked about these pumps. These are the pumps that pump 3 sodium out of the cell and 2 potassium into the cell. They are a mechanism of primary active transport because they're hydrolyzing ATP in order to energize this process. And so what we see here is that this pump is actively pumping sodium out and potassium in, right? And it's found on the basolateral membrane of our epithelial cells. We also have potassium channels, so these are leak potassium channels, and they're allowing potassium to leak right back out of the cell, and then leak sodium channels on the apical membrane allow the movement of sodium in. And then we also have glucose carriers that help to mobilize glucose. We'll look at an example later on, but also found on the basolateral membrane. They're going to move glucose through the cell and then into the bloodstream here. Okay. Let's look at the structures on the apical surface. So we have those sodium channels, those leak sodium channels here. And this basically means that sodium is coming in according to its concentration gradient. We also have sodium-linked glucose pumps. So sodium-linked glucose pumps is what type of transport? From that question we looked at earlier. Co-transport, perfect. So this is co-transport because both of our solutes, both of our molecules are moving in the same direction. They're coming into the cell. It's just that one is moving freely or passively according to its gradient, right? And the other is moving against its gradient. right? So the energy that is released as sodium moves along its gradient is used to energize glucose moving against its concentration gradient. Now, when we speak about this movement, we really want to differentiate this from what we talked about with GLUT4 transporters. So if you remember, we talked about the uptake of glucose from our cells. They used a GLUT4 transporter, which was a carrier protein, right, that facilitated diffusion. This is totally different from this scenario. All right, this is how we bring glucose across our epithelia. This is how we absorb it into our bloodstream. When we look at GLUT4 transporters, that's how it's uptaken into our cells from the bloodstream. All right, so students often confuse those two. So we have uptake into the bloodstream, and then our transporters or carriers uptakes it from the bloodstream into our fat cells and our muscle cells to be metabolized. All right, so we just remind ourselves here that this is co-transport. We have the movement of sodium and the movement of glucose with the sodium-linked glucose pump. All right, so let's look at the movement of sodium here. We talked about these two structures, right, the sodium leakage and the sodium pump, maintaining the relative ion concentrations, right? There's low intracellular sodium typically in the cell and high extracellular sodium in the body. And this is maintained because of the presence of the sodium-potassium pump on the basolateral membrane, as well as leak channels that are on the apical membrane. So what happens is sodium is being pumped out, it's going to go right around and leak right back into the cell. It's being pumped out, it's going right around and leaking right back into the cell. And so in that scenario, right, the way that that is set up, we don't have ever an accumulation of sodium anywhere. We don't accumulate it in an excess outside the cell, and we don't accumulate it in an excess inside the cell. And so we have very stable concentrations of sodium in the extracellular and intracellular environment because of these two channels, because it's being pumped out actively and leaked into the cell passively. Okay, this also happens with potassium. So we maintain our potassium levels because potassium is being pumped into the cell, but then it's leaking out passively. So it's being pumped in actively, leaking out passively. And so we don't have potassium accumulating either inside or outside of the cell. Those ion, I cannot speak today, those ion concentrations are going to remain stable on either side. Okay, let's look at glucose transport here again. So we talked about how glucose comes in. This is how it's being uptaken from our diet, from the food we eat. So think about it coming in your GI tract here. It's in your lumen, and it's moving against its concentration gradient, but it's being helped by the energy that's being metabolized from the movement of sodium. So sodium is moving according to its gradient. That's giving some energy. Glucose comes in, and now glucose is going to be this is facilitated diffusion. It's going to be moved through a carrier or a transporter into the bloodstream, right? And so this is passive, whereas this is active. So glucose has to make its way right through this cell into the bloodstream, and so it's going to go through the apical membrane actively, and then through the basolateral membrane passively, okay? It's going to use an active pump on the apical membrane and a facilitated diffusion, which is a passive type of transport on the basolateral membrane, okay? Let's look at this question here. In this scenario, where is the glucose concentration higher, in the ECF or in the ICF? When we talk about ECF and ICF, we're talking about the fluid under here. So is there more glucose in this cell, or is there more glucose in this fluid or in the bloodstream? Where's the glucose concentration higher? If this is passive diffusion, where do we have more glucose? Inside the cell, right? That is the gradient that is driving the movement of glucose out into the bloodstream, out into the extracellular environment. All right, so the movement of glucose happens from a high concentration to a low concentration, downhill, spontaneous, passively. All right, so let's wrap up here by looking at water transport. Now, solids are pumped into the extracellular environment in order to create osmotic pressure that can then pull those solutes across the membrane. All right, and I'll say that again. We have pumps on the basolateral membrane. They pump solutes out into the extracellular fluid, and those solutes then create what's called that osmotic pull. Remember we talked about that? that ability of the solutes, because they are higher in concentration, to pull water to them. And this is how we absorb water across our GI tract. This is how we secrete water as well when we need to. So this osmotic pressure is created by pumping some solutes into the extracellular fluid and allowing those solutes to create that osmotic pull to move water from our GI. Let's say we just drank a full glass of water. It's going to move right through the cell and into the underlying blood vessel. So that's how we absorb water. Okay. Let's talk about cystic fibrosis here, which is an interesting correlation. So this is basically the ability or the inability to create mucus, right? To create fluid mucus and to release water or secrete water to help lubricate our mucus. And so what we see is that there's disruption of these chloride channels, and these chloride channels are really important for creating this osmotic pull. Chloride is the solute that we pump across this membrane, and because there's more chloride, it creates that pull that draws water into the lumen. This is how we secrete water. This is how we create mucus or create saliva, create our fluid, body fluids. And so because we have a defect in this channel, in this chloride channel, we cannot secrete sodium, right? We cannot secrete chloride. We cannot get chloride out of the cell. And so we don't have enough solutes here to create that osmotic pull. And so we have a much thicker, viscous mucus than someone who has a proper functioning chloride transporter. Right? So because we cannot create that solute gradient, we cannot create the osmotic pull of water. And this usually affects the fertility of patients with CF because they do not have as thin or as slippery their body fluids, their saliva, their mucus, their semen is thicker, much more viscous because it doesn't have as much water as it should. And that's because the movement of water is disrupted. All right, so I think